Well, we managed to make it through 2023 without a major epidemic or pandemic. And as we get into the holiday spirit and look forward to the new year, some of us took a look back and picked out some of our favorite JCM papers published in 2023, which I will tell you was no easy task, given that there were many good ones to choose from this year. And personally, I had to look through all of the issues to pick a paper. But looking through them, it really never ceases to amaze me the variety of topics that we publish on in JCM, including unique things like the evaluation of a molecular assay for the diagnosis of Buruli or ulcers, to development of an EIA for detection of Taenia coproantigen. And then, of course, we publish on a lot more of our bread and butter things like evaluation of new assays for TB resistance. We had multiple studies this year on women's health diagnostics, evaluation of molecular assays for congenital CMV. And then I'm also told that there were some awesome AST papers published as well. So that's just wonderful. But today, like I mentioned, 4 of us JCM editors selected a favorite paper of ours from the past year, and we'll share those with you over the next half hour or so, all the while wearing ridiculous Christmas sweaters and or Christmas headgear, as is now our tradition. So if you're watching, you may want to turn on the video option for your viewing pleasure. So welcome to Editors in Conversation. I'm your co-host and JCM editor, Ellie Thiel. And of course, I'm joined by JCM editor-in-chief, Dr. Alex McAdam from Boston Children's Hospital, who, we should note, was recently elected as the next president-elect of ASM. So congratulations, Alex. How does it feel?
Thank you, Ellie. It feels just great. I'm very excited to be next president-elect and to do everything I can for ASM and for all of the membership of ASM.
That's awesome. I'm sure you'll do a great job. If not, we'll tell you.
You will. What's up with the voice?
What?
What's up with your voice?
I have sudden onset laryngitis, but hopefully, so apologies. But you'll have to bear with me. It'll get better. But,
or not.
Or not.
Viruses.
Yes, we're going to listen to Melissa's hoarse voice as well.
Yes, sorry.
But regardless of our voices, we hope you've enjoyed previous Editors in Conversation episodes. And we ask that you please subscribe and review this episode wherever you get your podcasts. This episode is brought to you by the Journal of Clinical Microbiology, available at asm.org/JCM and on Twitter or X at jclinmicro. Editors in Conversation is supported by the American Society for Microbiology, which publishes JCM. And remember that if you have a paper accepted by JCM and are an ASM member, you can get up to 50% off of the publication fees. Okay, so today we are joined by Dr. Trish Simner, return guest on the end of the year show. She is the director of the bacteriology laboratory at Johns Hopkins University School of Medicine. Welcome back, Trish. Thanks for joining us. And hopefully this time around, you and I are going to know what all the words mean.
I hope Alex has us prepared well. But yes, thank you for inviting me back. Last year was definitely a lot of fun.
Yes.
And I hope it continues.
It will. And this year, we're also joined by return guest, Dr. Melissa Miller, who's the Director of Microbiology and the Molecular Microlaboratories at the University of North Carolina School of Medicine. Melissa, thank you for taking the time to choose your favorite paper and joining us on the podcast.
Thanks for having me back after my 2022 sabbatical. I'm happy to be back on the end of the year show.
We are happy.
We'll see if I get invited back again.
Yes, I guess we'll see.
This is also a little bit of a reunion because, Melissa, you and Ellie were the very first guests on the podcast back when I was the solo host. It was the two of you doing an episode on COVID-19 diagnostics, which I'm pretty sure remains our most watched and listened to episode.
It's kind of amazing.
PTSD there, too soon.
Glad to have us back together and glad Trish is joining us.
I listened to that one intently. That was a good one.
Yes, It was good. So I think for those viewers or listeners, I guess, that can't see us, I think we have to do a little bit of a fashion show because we all really went out this year. So we're going to have the JCM podcast fashion show moderated by me. So we're going to start with Trish. Trish, if you'd like to stand. Trish is modeling for you a Christmas tree. She has Christmas tree headgear with a star on top. She has the garland, the little baubles. It's kind of like a frock almost, for those cold winter nights. Thank you, Trish. You're going to take the headgear off then?
Yes, I want to be somewhat taken seriously as I speak.
Okay, wonderful.
I find your tinsel distracting.
My what? My tinsel?
The famous Seinfeld episode, right? The tinsel was very distracting. As long as we don't have feats of strength at the end of this. It might be hard virtually.
So we're going to ask Melissa to stand up. Please model for us. Melissa, this time, is an elf. She has a lovely headpiece. Is that a Christmas tree headpiece?
It is a Christmas tree.
Yes, and she has kind of the gesture elf wear with a belt. It's really, really beautiful. And we have our solo male model, Dr. Alex McAdam, if you could please stand up. He is wearing a black sweater that says Merry Birdmas. Very, very festive and fits with the paper he selected today.
Yes, it is thematically related.
It is. And then I'll just model myself. I am wearing beautiful, sparkly reindeer antlers and a very sparkly Santa. And to be consistent, all of this is from my children's wardrobe.
Does Santa have sunglasses on?
Santa has sunglasses. Yes, it's a cool Santa.
Cool Santa.
Did he steal Rudolph's nose? He's got...
Well, yes, he did. Yes, he did. So that's what we're wearing. We hope you enjoyed it. Okay, before we dive into the papers, though, Alex and I thought it would be fun if each one of us came up with one word to describe and summarize 2023 for us, Clin Micro related, of course. We don't know what the words are that we each picked out. So we'll see. Let's see. Who should we start with? Alex, maybe we'll start with you.
All right, let's start with me. And before I say my little phrase, I want to say that I predicted before we did this that at least three of us would have the same phrase. And I'm going to back off that and say that I bet that two of us picked the same thing, at least two. So mine is machine learning and artificial intelligence, which we saw over and over, those two, in the titles of JCM papers about interpretation of genome data for predicting AST results, first mention of AST, and also for image analysis in clinical microbiology. So that's my pick.
It's a good one. Very, very accurate. Let's see, Melissa.
Well, I might have needed machine learning to understand the question because I didn't know we were picking something related to the journal, so I picked something more practical. I said short staff.
Oh, yes.
So that has been the theme in microbiology for many of us this year, sadly.
And you had a paper on staffing, I believe, recently or a few months ago. So
yes,
I think
so it was longer than that, but that's okay. Time flies when having fun.
All right, Trish, what did you pick?
I hate to say this, Alex, but I think you're going to lose. You better not have placed a lot of money.
Oh, no.
I will say that artificial intelligence and machine learning came across my mind, but I went with the encore of AMR. I feel like finally we don't have a virus to overshadow antimicrobial resistance. And so I felt like this
Now you've done it.
Yes.
12 days or 13 days left of the year.
Yes. So anyways, that was mine. It was encore of AMR, just being able to draw more attention to the important topic and not have some sort of awful virus, shadow the importance of that threat. So
that's good. That's good. Well, mine is not AMR.
Oh,
Mine is not machine learning.
no.
Staffing crossed my mind. But for me, it's actually syphilis. I feel like I've been drowning in syphilis.
So when I think 2023, I think syphilis.
I think it's a good pick. Syphilis,
Yeah.
sadly, is having a moment.
It's having a moment, and congenital syphilis in particular has increased 500% in the last 10 years. So it's a problem. So, cool. Well, Alex,
when does Alex lose?
I think so. What did you
I
bet?
guess I
feel
do.
like we should have bet with Alex. We could have.
Oh, I have to give up the editor-in-chief's position.
Oh,
in a year. Or year and a half.
Yeah,
A year and a half from now, I'll stop being EIC because of this moment.
Yeah. All right. Well, thanks for playing along, guys. So I think we'll dive in to our papers. And we will start with Trish. Trish, what did you pick and why?
Sounds good. Okay. So you further challenged me this year. I usually have to pick 3, and now I have to narrow it down to 1. So it may or may not surprise you, but I chose the paper entitled Interlaboratory Comparison of Pseudomonas aeruginosa phage susceptibility testing by Parmar and others as part of the Antibacterial Resistance Leadership Group. It was published in November. So as we know, with the emergence of multi-drug resistance and extensively drug-resistant bacterial infections, there's been a resurgence in the interest of phage for therapeutic management of patients. Phage, or bacteriophage, are viruses that infect bacteria. They're highly specific to their hosts, so that helps limit off-target effects on normal microbiota, while also requiring susceptibility testing of the phage against the bacteria causing the infection. Unlike conventional antimicrobial susceptibility testing methods, there are no standardized phage susceptibility testing methods and no validated interpretive criteria. This becomes extremely important as we're trying to assess the role of phage for therapy, and standardized phage susceptibility testing are required for clinical trials and for the guidance of treatment in compassionate use cases. So there's really this need to develop a standardized phage susceptibility testing method. And in the current paper, they evaluated the accuracy and reproducibility of the 2 primary methods that are currently available. The first is the double agar overlay method that assesses plaque formation as an indicator of phage activity. This generates a qualitative result as active, the formation of plaque, so the phage was active, versus inactive, no activity of the phage where there's no plaques present. This method is very laborious and requires extensive expertise and really can't be ramped up to high volume testing. The second method that's currently available and evaluated in this study is the liquid assays that assess bacterial growth or metabolism in the presence of a phage. In this study, they use a metabolism-based assay using the redox dye tetrazolium dye in the biologic OmniLog system. The good thing about liquid assays is that they may be performed at a relatively high throughput because you can be testing them in a 96-well format. So in this system, the activity of the phage is assessed by the inability to establish log growth of the bacteria relative to the uninfected control and how long it prevented log growth. And that it measures the delay in reaching log phase metabolic activity when a given isolate is challenged with a specific phage. And that time, that delay in reaching log phase is known as the hold time. And a hold time of 4 hours has been suggested to reflect phage activity. So in this study, they evaluated the accuracy and precision of liquid phage susceptibility testing at 2 sites and the double agar layer plaque assay at a single site. For 4 phages individually, as well as a cocktail, so 5 preparations total, against 145 Pseudomonas aeruginosa isolates. So this was a multi-center study, but it's important to understand that there were differences in the methods applied at each site. So site 1 performed triplicate testing over 3 days. So 1 replicate over 3 days. This required a new phage, bacterial inoculum, media preparation on each day of testing. Site 2 performed duplicate testing on the same day, on the same run, so that they were using the same phage, bacterial inoculum, and media preparation. So a little bit more standardized in the replicate testing on that at that site. And then site 3 performed agar overlay method in 1 replicate. So agreement was assessed in kind of a different manner than we're used to, where with phage susceptibility testing, there's no ground truth or reference method. So they use methods of comparing agreement, both assay agreement, intra-replicate agreement at the sites, and then comparing the agreement between the sites. And they use some sophisticated statistical methods that I will tell you broke my brain trying to wrap my head around them. So just to do the paper justice, I'm going to go very high level, but I'll guide you, the readers, to further study the statistical methods applied and how they assessed agreement in further detail. If you want to get a coffee, take out the manuscript and read it over in your spare time. So in terms of results, there was high agreement between the duplicate measurements made on the same run at site 2. So remember, that was the one that used the same inoculum and media for those replicate testing. And only satisfactory agreement among triplicate measurements made on the different days at site 1. So triplicate measurements were performed on different days, which is more of a rigorous assessment than the duplicate measurements performed on the same day. And testing on different days reflects issues that you might have in terms of phage storage, media preparation, the phage and bacterial inocula that can affect your results. So when you do it on the same day with everything the same, you get high agreement. However, if you repeat at the same site over multiple days, you only get satisfactory agreement. Unfortunately, there was a further lack of agreement when you compared the results between the 2 sites performing the liquid phage susceptibility testing. They had lack of agreement and low precision for all phages tested. So unfortunately, it's kind of a negative study, where among the 725 data points, so the 5 phage preparations and the 145 bacterial isolates, only 22% of the bacterial isolates showed agreement between all measurements, whether that be by the liquid assays or the plaque assay. If you narrowed further the agreement between at least one liquid assay comparing with the plaque assay, you could improve the agreement to about 61%, which is 89 of 145 different phage bacterial combinations. The plaque assay tended to favor increased phage activity compared to the liquid assays. So again, unfortunately, it was this kind of negative result where we weren't seeing good agreement or reproducibility of the current phage susceptibility testing methods across the sites and methods. However, there was an interesting finding in this paper that they did observe differences between the individual phage susceptibility results and the cocktail preparation. So in certain instances, they observed no activity of the 4 individual phages, but then as a cocktail, they observed activity. In contrast, there are some scenarios where they might have seen activity of one phage individually, but as a cocktail, there was no activity. So some synergy, antagonistic, and even neutral observations were made, which really the authors highlighted and show that there's the potential of the success of phage cocktails in the absence of individual activity. And so this is a promising potential of evaluating phage susceptibility against bacterial isolates. And the authors point out that this point deserves further study. So overall, unfortunately, the results from this study suggest that the current phage susceptibility testing methods that we have need further development to deliver reproducible methods to support clinical trials and compassionate use. And to date, there really is no standardized method that exists for us to be able to do that. So further work is needed. So that's all I got for you.
Thanks, Trish. That was good. I actually did have a question. I know, shocking. But we're kind of moving more and more AST, well, a lot of molecular testing for AST. Do you think, or did the authors talk about the potential of, looking for phage susceptibility by molecular means?
Yeah, that's a great question, and something I had been considering because I'm fascinated in like predictive WGS for predictive AST. And I'm like, well, can we predict that the appropriate receptor is there and predict activity? However, it's unfortunately much more complex than that. And I think we're going to need some sort of phenotypic method. And this is because you could have poor absorption of bacterial surfaces, the absence of suboptimal structures or the phage receptor. There could be blockage of entry of the phage DNA into the bacteria due to superinfection exclusion. So that would be a previous phage that's hosted by the bacteria that might allow binding of the second phage coming, but it won't allow the injection of the DNA into the bacterial host. There could be infection prevention by restriction modification, or even abortive infection where cell death occurs before the progeny are produced to allow for further phage activity. We did not plan that, but I kind of figured that. So I prepared for that one.
Wow. And I thought just like regular AST was complicated.
And that's another point the authors made, is that we know that even standard conventional antimicrobial susceptibility has its flaws. Now we have further biologic variability where we need to foresee this multi-step process of this phage infecting the host at various steps to actually see success for activity. So I do think this will be a challenging area that we're going to need to address. And in ClinMicro will be something that we might need to understand further as more standardized and reproducible methods are developed or created.
Nice.
I have a question for the group. And since I'm doing so well with predictions today, I'm going to predict that at least Melissa will say yes to this question. Have you been asked to send isolates for phage susceptibility testing?
Yes.
Yes, mycobacterial.
Yeah.
They tell me yes.
What'd you say?
I said they tell me yes, like Dr. Patel and Dr. Nobody has asked me personally to do that.
We have also, and the reason I said I thought Melissa would be likely to say yes is for us, it's mainly been in our patients who have cystic fibrosis, because those organisms develop pan resistance. And so we have to look for alternatives. I don't think we've ended up treating anybody yet, but we have sent a number of isolates for testing.
We have definitely treated people with M. abscessus with phage. To your point about have we been asked to send them out? Yes, we have, but we can't actually send them out because they go to a research laboratory, not a CLIA certified laboratory. So our hospital compliance got onto us. And so it goes through an IRB research route and a coordinator sends them to the research labs. I know this has been a question. I've had several of my colleagues ask me about this. So are you sending them out from your clinical lab, Alex?
We send them directly from our clinical lab. It is under an IRB approved protocol, and that's part of the protocol.
Same here. We also have the lawyers involved through material transfer agreements. So it is not a quick process in any way at this point. We have published on successful case of a extensively drug-resistant Pseudomonas infection that we treated with a combination of cifidrocol and phage. And so we have seen some successes. But, reading this paper really made me, reassess when we're sending it out, what is the accuracy of those results, thinking about, the lack of reproducibility that we're seeing here.
And it doesn't even get to the point of predicting the clinical response, right? We're not even to that point yet.
That's the next step. Yeah.
Awesome. Well, thank you, Trish. That was a great paper. I think next on the docket is Melissa. What did you pick?
Yeah, so speaking of mycobacteria, so I, like Ellie, did go through every single edition from 2023, and this one remained top of mind for me. So I chose from the July edition of JCM, the paper entitled Emergence of Inducible Macrolide Resistance in Mycobacterium chelonae Due to Broad Host Range Plasmid and Chromosomal Variants of Novel 23S Ribosomal RNA Methylation ERM55. That's a very long title, but it tells you almost everything that you need to know about the paper, even though the paper was quite eloquently done, and I'm happy to walk you through it today. So this was a collaborative paper. First author is Barbara Brown Elliott from the University of Texas Health Science Center, but there's also co-authors from Mayo Clinic in Minnesota and several institutions in Canada as well. There is an accompanying commentary that I recommend listeners and viewers also read. That was, I think, very insightful analysis of the paper. So most of our listeners should be aware that macrolides are cornerstone of therapy in patients with non-tuberculous mycobacteria, especially MAC and rapidly growing mycobacteria. Macrolides act by binding the bacterial 23S ribosome and inhibiting protein synthesis. So rapidly growing mycobacteria, which I will call RGM, hopefully that is okay with everybody, can be susceptible. They can be constitutively resistant, which is due to mutations in the 23S ribosome and RRL gene. They can be inducibly resistant due to the presence of a methylase encoded by genes. The most common gene that we worry about in RGM is the one found in 2 of the M. abscessus subspecies, and that's ERM41. And so to detect inducible macrolide resistance in most of our labs that are doing this testing, we either incubating the AST panels for up to 14 days, or we use PCR sequencing to more quickly identify functional versus non-functional ERM41 genes. However, neither approach is currently used for M. chelonae, as it was thought that this species didn't have inducible macrolide resistance. And in 2018, it was actually removed from the CLSI recommendation to hold RGM AST for 14 days. So I chose this paper because it is a very nice study that they did, but it also has very practical implications for us in clinical microbiology labs. It's important because it demonstrates that chelonae may be inducibly resistant to macrolides. Perhaps more importantly, from a big picture, it appears that this resistance is plasmid mediated, as they found it in other RGM species. So this has pretty significant implications when we start thinking about the ability of plasmids, particularly if they have resistance mechanisms on there, to be spread among mycobacteria. You'll find if you read the commentary, there's a very nice discussion about why really it's pretty rare for this, even though it's very common in Trish's lower order bacterial world to have plasmids swapping and inferring resistance mechanism. That's not very common in mycobacteria. There are a few examples that the authors mention in the discussion. So both of the reference laboratories here, UT Health Science Center in Tyler and Mayo, provide RGM AST for many, many laboratories around the country. So they have access to these strains that, once a phenomenon like inducible resistance in chelonae is identified, they can go back and kind of investigate retrospectively. So there are kind of 2 groups of organisms that were looked at. From 2018 to 2020 in Tyler, Texas, there were 5 isolates that had a clarithromycin MIC of 1 or greater at 3 days. So that's kind of the initial read of the RGM AST test. But then they had high-level resistance at 14 days, suggesting inducible resistance. Now, these isolates were found because of this higher MIC at 3 days. So they went back and retrospectively looked at these isolates at 14 days, and that's how they kind of discovered this inducible resistance to clarithromycin and Mycobacterium chelonae. There were 5 additional isolates from the following 2 to 3 years, 2020 to 2022. Of course, in my mind, I'm like, people were actually doing something other than COVID during these years, but I guess someone was. It wasn't me, but I'm glad that there was something else going on. So both Mayo and UT Health Science Center had started looking for inducible high-level resistance in Chelone because of these earlier isolates. And so this is greater than or equal to an MIC of 16 at 14 days. And so there were 5 more from these years, so that makes a total of 10 isolates. This number is important because we're going to break it down a little bit. They also identified 6 isolates that had intermediate MICs of 4 at 14 days. And I'm not going to talk about these more because they weren't this new ERM55 gene. There was no resistance mechanism that was postulated or found for them. But there's another group of Chelone isolates that will be interesting to look further into. So the 2 isolates from the same patient, one was smooth and one was rough. The smooth one was susceptible to clarithromycin. The rough one was inducibly resistant. Both of these were subjected to whole genome sequencing. So the resistant strain, the inducibly resistant strain, had an additional circular contig that was a presumed plasmid. And on this plasmid, it had a putative gene. And this was based upon 72% amino acid similarity to ERM46, which had been described from Rhodococcus. Now, if you look at its similarity to genes in other rapidly growing mycobacteria, it's less than 65% homology. So definitely a unique methylase gene. And so this newly discovered gene was called ERM55. It has a superscript P for plasmid, and that's because they also found other flavors, I guess, of this gene. So they developed a PCR assay, and the 10 resistant isolates, remember, for those 2 groups of years that had inducible resistance at 14 days were tested. And 6 of 10 of them were PCR positive for ERM55, but 4 of them were negative. So then these 4 were also sequenced, and they found ERM55-like sequences, but they were not on a plasmid. So there were, of these 4, 2 and 2. The 2 were identical to each other and were 86% homologous to the plasmid 55. The other 2 were homologous and were 82%. Were identical and were 82% homologous to the ERM55. One was found on the chromosome, so this was ERM55, and they used a superscript C to designate this finding. And then the other was associated with sequences that are usually found in transposons, so this gets the ERM55 with the superscript T to indicate transposon. So now they had these sequences, they could develop PCRs for the plasmid, the chromosome on the transposon version of ERM55, and test isolates for these ERM55 variants, as well as the mutations that we know in RRL, and also the presence or absence of a functional ERM41 gene. So only the 10 high-level resistant isolates at 14 days of AST were positive for ERM55. Everything was negative for RRL mutations or ERM41. So then they take these sequences, particularly the plasmid ERM55 sequence, and they blast against mycobacterial genomic sequences. And they found 2 isolates each of Mycobacterium iranicum and Mycobacterium abuense, both are rapidly growing mycobacteria, have the plasmid version of this ERM55. In table 3, if you go and look at that table, it tells you more about these isolates. And what you'll see is that the oldest one is actually from 2008. So even though this paper is describing a phenomenon that was identified as being reported from 2018 to present, this gene has been around for some time and in multiple rapidly growing mycobacterial species. So a few key takeaways from this paper is, we had previously underestimated rapidly growing mycobacteria in terms of developing inducible macrolide resistance. So this may be emerging. I think it's important that we start monitoring this. The plasmid that was discovered has elements such as type 4 and type 7 secretion systems, which takes me back to grad school. It's a little disconcerting to have to read about plasmids and secretion systems. But this, along with the discovery of the other mycobacterial species that were identified, the Iranicum and the abuense, suggests that this plasmid is transferable. But this needs to be proven. And this is one thing I really liked about the commentary, is they do a really nice job of reminding us about Koch's postulates and proving that finding this gene and having this observation of inducible clindamycin resistance actually still needs to be proven. The chromosomal and the transposon located ERM55 genes may also be mobile, but this is not yet known. And interestingly, the 10 isolates with the plasmid ERM55 of M. cheloni were from 8 different states. There were different types of infections, and so this is suggesting this is not a clonal event in one state or a couple of states, that this is likely more widespread. So finally, and this is for Trish, because I told her something this week, the TLDR is...
Thank you, Melissa. I now feel cool.
I know, you're so cool. TLDR, and everybody that knows me knows, like, I love bulleted emails, like, so it's perfect for me.
Yeah, Trish and I learned that this week as well.
We learned the
Do you
hard
want
way.
to define it for the listeners who may not know?
Too long, didn't read. TLDR is that the authors recommend going back to 14-day AST reads for Mycobacterium cheloni. At a minimum, they say, if the isolate has an MIC of greater than or equal to 0.5 at 3 days. So when I read this paper, immediately my lab had to create an online spreadsheet, and we start saving all of these data. We've actually not seen one yet be inducibly resistant at 14 days. We're looking at all rapidly growing mycobacteria, but the paper just reminds us, that things change in the AMR world, and mechanisms of resistance certainly are transferable and evolving. So that's all I have.
I'm
thought
so
that was a
glad
great-
you summarized that for me.
I'm here for you, Ellie. No problem.
I thought it was a great paper, and I particularly appreciated the commentary as well.
Yeah, it was really nice. It really took me back to my core science days of how do you prove a new finding. Very, very important observation and actionable still in the clinical microbiology laboratory. But there's still, and the authors point this out in their paper and their very nice discussion as well, that more work needs to be done on the plasmid, on these strains, and understanding what it means genetically.
It's true.
I'm adding the commentary to my list of coffee and manuscripts on Saturday mornings. What are your thoughts about communication with CLSI to kind of change the guidance there? Or do you think that some of these future studies need to be completed first? What's your standpoint?
Yeah, that's great. And as you know, Barbara Brown Elliott is, and in case she listens to this, I have to tell this story about her. I love her. She has been great to me in my career. And in the mycobacterial, especially AST world, I had a sticker on my computer for the longest time, which was WWBBEG. What would Barbara Brown Elliott do? I told her this one time. She is a hero to me. So everybody else on the paper too, but really, right, she has been very central in a lot of the AST work that has been done with Richard Wallace in Tyler, Texas. So back to your question, Trish. Sorry. CLSI, she actually mentions in the paper, they mention in the paper about going back to CLSI, and that would be the next step. I think that, our labs that, continue to want to be on the cutting edge and not necessarily wait for CLSI. It's not that hard to keep a tray to 14 days and just read it then, depending on your volume. That might be different in some of your labs. But for us, we're happy to go ahead and do it to see what we learn. It's still kind of in the research mode, trying to figure out what it means. So yeah, it will be interesting to see what comes out of the next CLSI meeting that discusses for mycobacterium.
Very exciting. I guess the last thing I will add is that I was super excited that there's representation from Manitoba, my home province. So
I actually thought of you.
I actually have Manitoba written on my notes.
Yeah, I thought of you, Trish, when I did this. And I thought about mentioning all 3 of the institutions in Canada. And I was like, I'm probably going to have to check on the pronunciation of English before I say them. So I just put them in. I know Canada's a very big country, but there were 3 institutions that were included, including the University of Manitoba.
I love it. Alma mater here.
Awesome. Thanks, Melissa. Let's see. Alex, what you got?
I picked something a little different. Ellie, you'll be glad that this is not about AST. There's about, 15 seconds worth of AST in this, but mostly it's other stuff. I picked whole genome subtyping reveals population structure and host adaptation of Salmonella typhimurium from wild birds. This is by Drs. Fu, McAnantha, and Dr. Dudley. Apologies if I have mispronounced any of those. And they are from the Pennsylvania State University and Pennsylvania's Department of Health. I chose these because it has 2 or 3 things that I love. It has evolution, and I have a little sideline collaborating with a group that looks at evolution of bacteria during chronic infections. It has birds, which I love, and it also has One Health. One Health, of course, being the recognition that human health, animal health, and environmental concerns intersect. So let's see. By way of background on Salmonella, the CDC estimates that there are about 1.35 million cases of Salmonella in humans per year, and that leads to 26,500 hospital admissions and 420 deaths. Most infections with Salmonella are acquired from food, often in meat or poultry or eggs. And another route of transmission, which is a less common one, is contact with animals or birds. There are large outbreaks among birds in the winter, and this has been linked to bird feeders, where the birds are congregating, and there is potential for transmission. And anyone who has bird feeders should keep track of the recommendations to clean those bird feeders to avoid spreading Salmonella among birds. But interestingly, there have been well-documented cases in humans that were associated with bird feeders. So there's a nice paper in Emerging Infectious Diseases, which described 30 cases of Salmonella in humans during 2020 to 21, with a genotype that had been found already in birds. Specifically, because I know Trish wants to know, it was a pine siskin, which is a finch. 64% of those people had a bird feeder, and 32% of those people had contact with a living or a dead bird prior to onset of illness. And then the same strain of Salmonella typhimurium was found to be widespread in birds in Oregon, Washington, and California. So that's by way of background. Let's start talking about the paper specifically. Salmonella serotype typhimurium is the most common species of Salmonella among birds. And this serotype is generally considered to be a generalist. And what that means for Salmonella is that it has a broad host range, which includes people, animals, and birds. And we can contrast that to Salmonella serotype typhi, which is a specialist, which has infections which are largely limited to humans. Each type of host, including different types of birds, represents a different environment or ecological niche for the organism. And just to take a straightforward example, antibiotics are frequently given to humans or to agricultural animals, but not to wild birds or wild animals. So adaptation to the specific niche may be important for the bacteria to cause infection. Beyond antibiotics, there are also questions about the nature of the immune response in the host and the microbiota that the organism finds itself in as well. So in this paper, they asked the questions, first, are there specific lineages of Salmonella typhimurium which have adapted to specific groups of birds? And second, when did those lineages begin to adapt, if they exist? And what type of evolutionary adaptations did they make to that host? So they started with 787 Salmonella typhimurium isolates that had been collected between 1946 and 2021 from 18 different countries. Most of them were from the United States, Europe, or Australia. And the whole genome sequencing data for these 787 isolates was in a database called Enterobase. The authors contributed 414 of those genomes, and the rest were contributed by other investigators. The ones that they contributed were all from the United States, and they were sequenced from birds that were sick were found dead. They broke the analysis down into 5 different groups of wild birds. We're only talking about wild birds, no domestic birds. And I'm going to use common names for these groups. There were some isolates from other groups of birds outside of the 5 that I'll mention. And there were lots of isolates from bird species where the species information was not available in the database. So the 5 groups that they included were, first of all, the songbirds or the perching birds, including things like sparrows and cardinals. Second, the seagulls and terns. And if you don't know what a tern is, you should pause and Google it. The third group was the ducks and the geese. And the fourth group were the pigeons. The fifth group were a group of other water birds other than ducks and geese. And that includes pelicans and herons, which are part of a clade. Now, I can see Ellie wants to ask me, why were pigeons pulled out as a separate group from the other perching birds? And the answer is that these groups of birds all split from each other 65 million years ago. And that's when an asteroid hit the Earth and there was a massive extinction event, the Cretaceous-Paleogene extinction event, in which 3/4 of the existing species, including all non-bird dinosaurs, birds are dinosaurs, including all non-bird dinosaurs, went extinct. And that was followed by a massive diversification of the remaining creatures, including the birds. So that's when these groups split apart.
I am just going to add that none of that was in the paper. That was all just from Alex.
I did a lot of reading about this. I went into some of the stuff I really went down the rabbit hole. Okay. So here's what they found. And don't ask me about the methods. Trish can talk about the methods. She knows much more about this than I do. I don't understand the data analysis. I am a consumer of these data. I am not a producer of these data. But I thought that the results were absolutely fascinating. So first of all, they used the whole genome sequencing results to form a nearest neighbor tree using whole genome multi-locus sequence typing, or MLST. And they used over 21,000 loci for that MLST. Then they used a filtered subset of data where the data included good metadata, the date that the bird was collected, the location the bird was collected, what type of bird it was, to do some core genome SNP-based phylogenetic trees. And I'll go ahead and present those without differentiating much between the two of them. So first of all, the Salmonella typhimurium from the 5 groups of birds fell largely into 7 different lineages. There were 2 lineages from the songbirds, 2 from the pigeons, and 1 each for the other groups, the seagulls, ducks and geese, and the other water birds. Each of those lineages was very strongly associated with that specific group of birds, meaning that the lineage was only rarely found in the, quote, wrong group of birds. One thing that I thought was interesting, which the authors did not call out, was that in several cases, when the Salmonella typhimurium was isolated from the wrong type of bird, it tended to be isolated from a bird of prey, such as an eagle or a hawk. And it made me wonder whether that eagle or hawk had been feeding on one of the birds in the group. Like it got the pigeon strain by eating a pigeon, for example. All right, going on. There were also 3 different lineages of Salmonella typhimurium that were isolated from diverse groups of birds. And those we might consider to be generalists within the bird population and perhaps other hosts as well. Neither time that the isolate was collected nor the geographic site where it was collected were associated with the different lineages. And that suggests that those lineages really have evolved to be specific to that group of birds, and they've evolved to succeed within that group of hosts. The core genome SNP-based phylogenetic tree was consistent with all of this. One thing of note, which I think is pretty well understood at this point, is that there's a commonly used typing method, sequence typing, in Salmonella, which is based on MLST of just 7 housekeeping genes. And when they used that, it was not adequate to differentiate between these lineages of Salmonella typhimurium, which is not surprising, as I said. It combined lineages that were differentiated by the larger whole genome sequencing. All right, so next question they asked is, when did these lineages emerge? And to me, it was surprisingly recent when these lineages emerged. 4 of them emerged around 1950, between 1943 and 1969. 2 of them emerged earlier, one in 1926, and that was the duck-goose lineage, and one in 1847, which is the pigeon lineage 2. It does not necessarily mean that Salmonella wasn't infecting birds before that, because there may have been lineages that were infecting the birds before, and they were replaced by these lineages. There is a pangenome analysis, which I think you should look at if you're interested in it, but I am going to skip it in the interest of time and go right on to the data about antimicrobial resistance. All of the antimicrobial resistance data are predicted from the whole genome sequencing data. And the bottom line is that these bacteria have very few genes that would mediate antimicrobial resistance. They all had one cryptic, meaning unexpressed aminoglycoside acetyltransferase, in all of them except the duck goose lineage. The duck goose lineage had 2.4 genes on average, where the others only had one, and that was that acetyltransferase that I mentioned. And you can compare that to the isolates from humans or domestic animals that they also included in their analysis. Now, humans, of course, and animals, domestic animals, get antibiotics, and there they saw from 2 to 8 resistance genes rather than the 1 to 2 that they saw in the birds. They looked also at whether or not there were plasmids, and they looked at the presence of virulence genes in these isolates. And I'm going to combine those because there's a common theme between them. So first of all, one of the lineages in songbirds lacked 2 plasmid replicons, suggesting that they lacked the plasmid itself. The other songbird lineage lacked these replicons in 40% of the isolates. This is for a plasmid called PSLT, which is an important virulence plasmid in Salmonella that encodes genes which are needed for invasion and intracellular survival of the organism, among other features. These 2 lineages also had fewer genes associated with virulence than most of the other lineages, which is not surprising because those genes are on the plasmid itself. But on top of that, other virulence genes, not on these plasmids, were absent or they had mutations in them in all of the different bird lineage Salmonella. There were a lot of mutations in the genes for type 3 secretion systems. Type 3 secretion systems make these little needle-like projections that can inject factors, proteins, into host cells that alter the function of the host cells. There were also mutations in genes for extracellular matrix proteins that are needed for adhesion and for biofilm formation, as well as the genes for Fimbriae proteins. So all of these genes for virulence factors were mutated, and in many cases, they would lose function. So taken together, these results suggest that so-called genome degradation, or probably better called genome reduction, is an important mechanism of adaptation in Salmonella typhimurium to the bird hosts. That is a form of evolution where genes are lost. And we think of evolution as organisms gaining new functions, but in fact, when organisms find themselves in a new environment or are facing specific challenges, they will sometimes evolve by actually shutting genes off. And an example from humans is the loss of the Duffy blood group antigen on red blood cells in most of the populations in West Africa. And the Duffy blood group antigen is a receptor for Plasmodium vivax. So loss of that antigen is a way that people can evolve resistance to infection by Plasmodium vivax, a cause of malaria, of course. So this is I thought this was absolutely the fantastic paper. I loved everything about it. The TLDR version is, yes, the bacteria did adapt to the host environment. Yes, there were specific populations of bacteria adapted to specific groups of birds. And the most interesting thing to me was this loss of genetic function or loss of genes as part of that evolution. So I will stop there because I can see that all 3 of you have a lot of questions.
Well, I appreciate the TDLR, let me tell you. I did have one question. How did they collect the samples?
They don't say.
They don't say.
They don't say.
Okay.
Probably with a swab.
Are they live birds or dead birds?
Both. The ones that they sequenced were from diseased birds as well as dead birds.
Okay. Awesome.
I, of course, picked up.
Go ahead, Trish.
No, I was just going to say, of course, I picked up on the AMR finding. That I thought it was fascinating. And, the selective pressures of pushing AMR and the number of AMR genes harbored by Salmonella versus the not domestic birds, wild birds, that Alex just talked about. And then the other thing is like strong observation, Alex. I did note some uncolored circles in the wrong lineages. And I was wondering, I'm like, oh, that's odd. But the prey bird observation was really good. Congrats.
Thanks.
You should reach out to the authors.
That was really nice. Thanks, Alex.
Yep.
Well, I guess we'll turn to my paper, which Trish, you were talking about like breaking your brain. All three of yours broke mine. So this is a much more basic, still good, but much more basic paper. So I chose a study by Johannes Traeger and colleagues, which was done primarily out of Germany, and it's entitled Detailed Beta-1,3-D Glucan and Mannan Antigen Kinetics in Patients with Candidemia. And there are really two main reasons I chose it. First, I thought the authors did a really nice job doing a deep dive in and looking at the kinetics and fluctuations of beta-D-glucan and Mannan levels in serially tested patients. So they looked at over 1,200 samples in their patient cohort. And then more importantly, they outlined specific clinical scenarios for when doing serial antigen monitoring would be of clinical use. And then my second reason is much more selfish. So I am not infrequently asked about what the role is of Mannan antigen testing as an alternative way to diagnose candidemia. And I think this paper really nicely showed the limitations of relying on that assay. So as a quick refresher, we all know that candidemia is a problem, particularly in the ICUs, where about 10% of bloodstream infections are due to candida, and those infections are associated with a mortality rate of roughly 30%. Typically, antifungal treatment is started about around the time of blood culture positivity, which is usually about, a couple of days after symptom onset. And as we all know, kind of that delayed initiation of appropriate treatment is associated with higher mortality rates. So as a result, there continues to be interest in identifying candidemia in those high-risk patients prior to symptom onset or blood culture positivity. And so one approach to do that has been to preemptively evaluate patients for the presence of various fungal biomarkers. So in this study, the fungal biomarkers that the authors really focused on were the Candida Mannan antigen and then beta-D-glucan, which we all know is this pan-fungal antigen found in the cell wall of most fungi. And so very briefly, what this group did was undertake a prospective observational study over a 36-month period where they collected residual serum from about 129 adults with blood culture confirmed candidemia. They measured beta-D-glucan and Mannan antigen levels in all of those samples, and then they evaluated results relative to when the samples were collected. So either before the first positive blood culture, near the time of the positive blood culture, and then more than 3 weeks after that candidemia episode. And very, kind of getting to the point, because it was a really, to the point study. They had two main findings that are important. First, they showed that beta-D-glucan was detectable in 63 to 78% of patients prior to or at the time of the first blood culture positive event. And that's compared to about a 35% positivity rate of the Mannan antigen in those same patients at the same time. And one reason for that lower Mannan antigen positivity is because not all Candida mannans are created equally. The monoclonal antibody that's used to detect Mannan actually does not detect the Mannans from Candida parapsilosis at all, and it does not detect them well from Candida krusei either. And so those two species accounted for about 13% of their candidemia infections. And so based on these data, the authors really concluded that testing for Candida Mannan really has no role to play for early detection of candidemia. So that's number one. The second key takeaway, they looked at the kinetics of both beta-D-glucan and Mannan antigenemia during treatment to see if the trends could be used to predict recovery or relapse. And just for the purposes of today, we're just going to limit talking the discussion to beta-D-glucan because of that limited sensitivity of the mannan antigen. And so what they showed with beta-D-glucan is, they couldn't identify any specific thresholds to indicate recovery or relapse, but they did show that downward trending of beta-D-glucan measured every 3 days to weekly following initiation of treatment was associated with recovery in 85% of patients, whereas on the flip side, increasing antigen levels were associated with fatal outcomes in 73% of individuals. And then they also showed that an increase in beta-D-glucan levels of 290 picograms per ml or higher from the previous value was associated with a relapsed candidemia event in over 2/3 of patients. And so based on all of that, they recommended daily beta-D-glucan monitoring in patients without clinical improvement following initiation of therapy, where 2 consecutive BDG level increases or an increase of 290 picograms per ml or higher should trigger additional actions, including a more in-depth workup or a change in therapy. Whereas for patients that are improving, they specifically state that additional serial testing is not beneficial. So bottom line, there's really no need to do, just to check testing or testing just to see how things are going, which unfortunately is not uncommonly done. So again, I think this study is useful because they included a really large number of patients with anywhere from 3 to 27 serial sample draws to look at kinetics. They also showed the fairly limited utility of mannan detection. And then whether you're on a, love beta-D-glucan or hate it, because there's nothing in the middle, you either love it or you hate it. It can be used as an early marker for detection of invasive fungal infections and for candidemia. And it can have a role to play in as a prognostic marker for many patients. So that's it. Short and sweet for me.
I have a question, Ellie.
Yeah.
So the pre-blood culture data, right? So the idea and part of the problem is diagnosing candidemia early and fast. So what are your thoughts based upon these data? Like, how does this get implemented clinically? Do you identify high-risk patients for candidemia? Are they getting the just check beta-D-glucan daily while they're in-house? Like, I happen to be in the mostly hate beta-D-glucan fan club. Although I do like it for pneumocystis, but that's why I said mostly hate. Maybe there's a spectrum. What do you think about that in terms of using it in that role?
Yeah, so it's tricky. I think, they stress the fact that it's, after you start antifungal treatment, if there's no improvement in the first 24 hours or so, they would recommend, looking for trending after that time point. So that's kind of their recommendation. I don't know that that's how it's done, though. In my experience, people start, don't improve, and they do a beta-D-glucan test, see that it's positive, and that's it, one and done, But I think the key is trending it. And I thought it was nice they show, either daily or every 3 days as an implementation. But yeah, the use of beta-D-glucan, I think, is all over the place. At this point, there's no guidance.
Yeah, I can see some physicians looking at figure 1 and saying, 3 days before 10 to 15% had a positive beta-D-glucan. So I should daily beta-D-glucans on my patient.
Yeah. And I think the other challenge is the issue of analytic specificity with beta-D-glucan. So you really have to know what your patients have been given by IV, whether they've had recent surgery, because all of that is going to impact how you interpret the results. And it's 2 different things, right? It's identifying a candida infection before symptoms occur, so the prognostic, and then the post-treatment. So it's 2 separate things. So there you go. Solve the beta D-glucan problem.
You solved the mannan problem.
That's what I was about to say.
Yeah, so nobody asked me what I think about mannan.
We need to work on the next generation one.
Yes. Awesome. Well, I think we did a great job, if I don't say so myself.
I agree.
Thank you, Trish, for agreeing to come back on the podcast again. I think we managed to get through it without any major faux pas.
I think we did fairly well. Thanks for having me back. Always a joy.
Yes, and Melissa, thanks for coming too.
Yep, thanks for inviting me. I'm gonna go home and clean my bird feeder now.
I think the question on the table is whether this podcast should pivot at this point. We should focus on birds going forward. Oh, look at Ellie's face.
Okay, Alex, when you first asked me to do this, I said I don't do AST. And I'm going to modify that statement. I don't do AST or veterinary papers.
See, right. Ellie hates birds. That's what I heard.
TLDR, Ellie hates birds.
Flamingos.
Okay, we got to end. Oh my gosh, this was great. Alex, I think it was a good episode.
I think it was a tremendous success.
Yes.
Happy holidays, everyone.
Happy holidays.